At this point in the course, you should be very familiar and comfortable with this idea of natural selection, right? Which is the idea that certain traits can be beneficial or detrimental. You should also remember that whether or not a trait is beneficial, right, whether it's an adaptation or not, is completely dependent on the environment. So in an environment with a very light substrate, these mice with lighter coats, that's the adaptive trait. So the adaptation is to have a lighter coat. The darker coats don't do as well because predators are more able to see them. We go a couple dozen miles away and we may have a volcanic dark substrate. And in that case, then suddenly the light coat is no longer an adaptation and the darker coat is the adaptation. So again, just remember. Now, another thing that can play a very important role in the evolution of these traits is the genetics. So we're going to look at several examples. We'll mostly stick to the fairly simple dominant recessive, but we might look at some examples on the simulator of some codominance and a couple other things that occur. So, you know, we could use big A, big D, little d. In some cases, you'll see A1 versus A2, and those are used for the different alleles. So just be aware. So in this case, we've got for the dark coat, big D is a dark individual. Big D, little d is a heterozygote. They may also have a dark coat if D is dominant, and the light coats might be little d. And so we might have different trends in different environments depending on the genetics of this situation also. But what we could do is experimentally go into the environment and test out the relative fitness. So the relative fitness of this big D genotype is going to be dependent on the environment. In a dark substrate environment, the fitness level of the big D individual is going to be, by definition, 1, right? Because that's as fit as you can get. And then lighter coated individuals are going to have a somewhere in the lower one. But remember, in a light one, then the fitness of the big D individual may be somewhere lower than 1, and this individual might have the relative fitness level of 1. And then these other ones are going to depend on genetics. So I just want you to think about, there's no straightforward, clear answer to this. It's a complicated answer for which we may need to go into the field. We might need to gather data. We may need to also evaluate the genetics. Is it complete dominance? Is it codominance? Or are we seeing some other more complex genetic feature playing a role here? And we're not going to get into the really complex ones, but we will look at dominance versus recessive alleles here in a little bit. But once we do all that complicated data gathering and analysis, we could then perhaps modify the Hardy-Weinberg equilibrium to take into account this effect of natural selection. Now, for the purposes of this course, we're not going to get into all the details of that, not do it in any real quantitative manner, but I do want you to evaluate it. So you know what the Hardy-Weinberg equation is, right? So I'm going to put it up here. I'll just type it again. I'm going to have to go back and put in the superscripts. So p squared plus 2pq, hopefully this is getting familiar and comfortable for you, plus q squared. And again, this is always going to equal 1, but we're going to compare our predictions to our original observations to see if they balance. Okay, so let's go back to this. All right, so here we are. P squared plus 2pq plus q squared. Now that's the regular one. And if we came up with an imbalance, then we would maybe need to add something to this equation to balance it back out. Now, it's difficult to determine whether that imbalance is determined by natural selection or genetic drift or new mutation or migration or non-random mating. We would need to do some research and some data gathering to figure that out, right? But if we figured out it was natural selection, we could perhaps adjust it. So for instance, if we found out that one allele was being removed from the population by natural selection because it was detrimental, we may adjust this equation. So you may see something like this. So p minus s squared, and that s is the coefficient of selection. And then you might see something here also where we put the s in this part of the equation also, if that is also an impact in the heterozygote. And so we have a modification of the Hardy-Weinberg equilibrium to help put everything back in balance, and this helps us determine the impact of natural selection. Okay. Now again, we're not going to do this in any quantitative measure, but if you see an s in the Hardy-Weinberg equilibrium, know that that is an adjustment for natural selection, and it's called the coefficient of selection. And that's as detailed as we're going to get for the purposes of this course. Now, The type of natural selection that we've been talking about to date is more precisely known as frequency-independent natural selection, but that's a mouthful. So what we're going to do is just refer to it as regular old natural selection. And what we mean by the frequency-independent part is that your fitness is not determined by how common your trait is in the population. For example, let's say that having a dark coat is good and your fitness level is 1 because you live on a dark substrate. That fitness level is going to stay the same no matter how many other individuals in your population also have the dark coat. That's the best fitness level. It's not going to be modified at all. Okay, so your fitness does not depend on the frequency of the alleles in the population. And that's the most common and simplest type. But genetics can begin to play a role in some cases. And so let's look at the role of genetics in frequency-independent selection. And then in the next lecture, we'll talk about the frequency-dependent version. To do this, we need to understand the process of natural selection and its inevitable result. So the inevitable result of directional selection, which is one type we're going to talk about, is fixation of one of the alleles. Now, fixation simply means that one of the alleles is removed, the detrimental allele is removed, and the other allele is fixed or fixated in the population. It goes to 100%. So fixation, right, means you lose one allele and you gain, and the other allele goes to 100%. And so the Hardy-Weinberg equation is really nice and simple once the allele has become fixated in the population, right? We would see something, probably cutting and pasting is a little faster. We would see something like this. Let's get rid of our coefficient of selection and go back to our regular Hardy-Weinberg equation. Right? P squared plus 2pq plus q squared. Right? But once an allele becomes fixed, now remember, if we're going to complete our Hardy-Weinberg equilibrium, we're going to put this equals 1. Oh man, this is still a superscript. It's going to bother me, so let's change it back. Okay. Let's move that down here so it's a little easier to see. So that's our regular Hardy-Weinberg equilibrium. But if we lose one of the alleles, meaning the p-value, let's say, goes to 1, the q value goes to 0, right? So suddenly 2pq becomes 0, q squared becomes 0. And so the Hardy-Weinberg equilibrium, once you've reached fixation, is easy. Right? It's simply p squared equals 1. And p is the value of 1, 1 squared is 1, and that's simple. And now once we have fixation, there's no more possible chances for natural selection, because if you only have one allele, everyone has the exact same fitness level. There's no diversity in the population for that gene, and so there's no natural selection that can work on it. The other forces of selection could still play a role. We could still have new mutations. Well, actually, I take that back. The other forces of natural selection could only play a role once mutation brings in diversity. So mutation can still be a factor in bringing in new diversity, and when it does, then all those other forces of evolution can take over. We can have natural selection, we can now have genetic drift again, but when it's fixed, we're stuck at that place. There's going to be no evolution for that allele, that gene. Other genes could evolve because they're not connected to one another because of random assortment. Okay, so just keep that in mind. You need to know the definition of fixation, and once genes have become fixated in the population, then essentially for that gene, evolution stops until a new mutation brings back in some diversity. Now, we have been looking at the two allele versions, but we can also have much more complex traits. So when we see a trait like this that varies continuously over the spectrum of traits, now this one we show 5 different color variations, but in reality, many times you see these traits that have these distributions that look like this. So height in humans is like this. You have some few individuals that are very short, some few individuals that are really tall, but most individuals fall somewhere in the middle. This is called a normal distribution or normal curve. And so when we see that, there are 2 reasons that contribute to characteristics that distribute this normal distribution. And that is that number one, it's usually controlled by complex genetics. Polygenic, meaning many, many genes all contribute to that same trait. And number two, environment might be playing a role in creating some variation and diversity also. Okay, but even if we have this, we can still have natural selection. So if it was bad to have the light colored wings, you would still see a shift towards more and more dark colored. But because of the complexity, it might take many hundreds of thousands of generations to lose the diversity that we're seeing in the population, because it's a much more complicated thing than just simply two alleles. And so we don't really have the same rate or the same speed of fixation in these more complex traits. And what you realize quickly is that most traits are more complex. Mendel on purpose picked very, very simple traits with one dominant allele, one recessive allele, and that's how he figured out the basics. But most genes are more complicated than that. Okay? But directional selection, even in complicated situations, would eventually lead to fixation of one allele. Now, directional selection is only one type of natural selection. So there are actually three general types that we are going to look at. Directional selection leads to fixation of one allele or the reduction of diversity in the short term for these very complicated situations. But we can also have overdominance. Okay? Overdominance also is known as heterozygote advantage, but that's a less common term and we're not going to use it. I put it there just in case you've heard it before or if you see it in other places, but we're just going to use the term overdominance. Overdominance is the name of this when we have a two allele system. But in a more complex system, we call it stabilizing selection. So you need to know the term overdominance, and that in the more complex examples, it's referred to as stabilizing selection. The most famous example, and the one you may have seen before in other courses of overdominance, is this example of the sickle cell anemia and malaria relationship that we see in humans. So this is how this works. Sickle cell allele is a detrimental trait, and it's kind of in this codominant situation. So if you have two sickle cell alleles, you usually die before you reach reproductive age. It's a very bad condition to have. However, if you have one sickle cell allele and one wild type allele, then you see a few minor side effects of carrying that trait, but it's not fatal usually. So not as huge a deal, but it's still not as good as having no sickle cell alleles, unless you're living in an area where malaria is very present. And this is what allows this to have an overdominance effect, is that the sickle cell trait means that you are resistant to malaria. And so if you're living in an area where malaria is a big, big concern, then you have this very interesting fitness level. And so what it means is that if you are homozygous for the wild type and living in an area with malaria, you don't do as well as the heterozygotes because you suffer more from malaria. So malaria reduces your fitness. The heterozygotes do very, very well because they are resistant to malaria and they see only minor impact from the sickle cell disease. The sickle cell homozygous, if you carry two alleles of the sickle cell allele, you don't do well at all. Again, you usually die. So usually this shows a fitness level of 0.8, but usually your fitness level would be much, much lower if you're homozygous for the sickle cell allele, because you usually die before you reach a reproductive age. But what this means is that the force of natural selection is balancing things out, because the heterozygotes are best, and the only way to make heterozygotes is to maintain diversity of these alleles. Eventually, we reach a balance point. Now, we'll look at this again a little bit on the simulator that I introduced to you at the very first part of the semester. So just for now, know that sickle cell malaria is an example of overdominance, and that the end result of overdominance is this balancing effect where we maintain both alleles in the population. Now, in more complex things, and we see this all the time in species, we call it stabilizing selection. So let's just look at one example in humans of stabilizing selection. Birth weight is actually a good example. So if you are born very, very small, you have a low chance of survival because you don't have as much fat on your body and you don't get as much of a head start. Now, we've reduced the effects of this somewhat with modern medical intervention. But still, having low birth weight means lower survivability. Having really, really high birth weight means low survivability also. And again, we've alleviated some of this with medical intervention, but really big babies have trouble with birth. There's more stress. Sometimes they even get stuck, and both mother and baby's lives are threatened. And so there is kind of this Goldilocks zone, if you will, not too big, not too small, but just right in the middle for birth weight. And so the result of that in the human population has been this stabilizing selection, where we see a few babies, but not very many, that are very, very small. We see a few babies, not very many, that are very, very big, and most babies end up somewhere in the middle. And so natural selection has worked to stabilize birth weight at that kind of optimal stage. We also have an interesting condition called underdominance. Now, again, the one in parentheses there, heterozygote disadvantage, you might have seen it. We're not going to worry about it. We're just going to use the term underdominance. An underdominance is what occurs in a two-allele system where the heterozygote has the lowest fitness level. So it's good to be homozygous for the A1 allele. It's good to be homozygous for the A2 allele, but it's not good to be homozygous, to be a heterozygote. And so what this ends up with is we actually, now if you think about this, what would happen, you might think, oh, that's going to maintain diversity in the population. But in actuality, it doesn't. And there are two possible outcomes. We either fix one of the alleles, and it depends on where you start. If you start on one point of where this balance line is, you will fix the A1 allele. If you start with a frequency that's lower than that, lower than this balance point, you would fix the A2 allele. So one end result of underdominance is fixation of one allele or the other, depending on the balance. The other one, which can be very, very interesting to science, is we could have a separation of this population into two distinct populations. And so think about this. If we have one allele, in this case the NZB allele is good if you are homozygous for that. The NZW allele is good. This is an example in laboratory mice, but there are natural examples also. These guys do really, really poorly if they have one of each of the alleles. But if white mice and black mice were in separate areas, or if there was some sort of mating preference where black mice mated preferentially with other black mice and white mice mated with other white mice, then we could potentially set up two distinct populations, one that was like this and one was like that. And this is very interesting to scientists because it can be one of the forces that helps to break a population into separate, distinct populations. And that long term might even result in two different separated species. And so underdominance is interesting because under the right situations, we might end up with some force pushing towards speciation. It could also just mean that if they're competing directly against each other, that one is better than the other. And so in this case, the A1 allele is better than the A2 allele, so there'd be some force for pushing these guys to extinction if they had to compete with those guys. And so in the more complex situations, this is called diversifying selection because, again, it can result in two distinct diverse populations. Okay, so let's look at a summary of all of those. Oh, so additional forces in place. We might have geographic separation. We talked about mating barriers. There might be mating barriers. But if there's some additional force, then we can maintain both populations if they're not competing too heavily against one another. So here's a summary of all of these types. And this is looking at the complex systems, not just the simple two allele ones. So we have directional selection, which can push a trait. Now realize directional selection can go either way. So if this represents heights in humans, and suddenly there's a big advantage to being tall, and tall people do better or reproduce at higher rates, then we'd see a shift in the population towards the taller phenotype. But we could also go the other direction. If instead being short was good and all the selection pressure is up on this side of the tall people, then we would see this directional shift going the other way. But we start with some distribution and it moves one way or the other. That is directional selection. And we call it directional selection in the simple two allele version easy. So that's an easy one to remember. Now, if there is selection on either extreme, so if it's not good to be on one extreme and not good to be on the other extreme, but individuals in the middle do best, then we end up with this stabilizing selection. And in the two allele version, this is called overdominance. And the way I remember this, if you want to remember it, is this kind of makes this hill, it makes the hill a little more pronounced and steep, and you have to go over the hill. That's how I remember stabilizing selection, overdominance. If that helps, great. If not, Just remember that stabilizing selection is a more complex version of overdominance. And then finally, diversifying selection. And diversifying selection can result in these two distinct subpopulations, maybe with some individuals in the middle if they're still interbreeding some. And again, diversifying selection could eventually separate these if we had other factors maintaining them, could separate them into two populations. Now, the two allele version of this is underdominance. And so you remember it makes this little U in the middle, this little valley, and that's underdominance. If that helps you, great. Okay? So those are the modes of selection. Now, a quick reminder before we look at some simulations of these things, that an adaptation is completely conditional. It completely depends on the environment. So if I ask you, say, having a loud mating call, let's say we're looking at frogs out in the wild calling for a mate, is having a really loud mating call an adaptation? Now, hopefully in your head you're saying it depends, right? Now, you'd say on the outset, yeah, that's a great thing, because then you get more mates coming in and you do better overall. So having a loud mating call is great. But what if there are lots of predators, and those predators are listening to that mating call? So here's an example. This male frog is sitting up on a rock, just singing his heart out, hoping that he will find lots of love, right? Oh, I don't know if frogs have love, but at least lots of mates, right? But instead he calls in a predator. So in an environment where predators are very common and your mates are relatively close, then individuals with less volume that have quieter mating calls, they're going to do better. But if you're in an environment with very, very few predators and maybe mates are widely spread over the environment, then having a very loud mating call is going to be better. So remember, adaptations are completely conditional, and we need some information about the environment if we're going to be able to decide. And we might even need to do lots of observations or experimental manipulation to make a final determination. All right, so I've got a link here in the PowerPoint to this evolution simulator. So we're going to take a look at that in a little bit. But the last thing we do before we look at that is I want to fill in this chart. So remember, the inevitable result of directional selection is fixation. But there can be many factors that determine how fast we reach the fixation point. So here I have 3 separate simulations run under different conditions. One of them reaches fixation after about, if this is 100 generations, after about 30 generations or so, we reach fixation. These other 2 have not reached fixation even after 100 generations. Now, we've greatly reduced the frequency of this big A allele, and the little A allele has much higher frequency, but we haven't completely reached fixation yet. But in this last simulation, the blue one I did, we reached fixation. The little A allele is now the only allele in that population. So there are 3 main things that determine the rate of fixation, okay? Let's add to this. Number 1, the first thing to determine the rate of fixation is the initial frequency. Let's make this a little smaller. The initial frequency of the alleles. So if we start at 50-50 versus starting at 0.2 and 0.8, we're going to have a different time period before we fix one of those alleles. And remember, populations could start out in balance, and then there could be some environmental change that could now push one allele so it's no longer good and begin to remove it from the population and fix or fixate the other allele in the population. The other thing that we can look at is the strength of natural selection. Now, we'll look at all of these in simulations here in just a little bit. And then the final thing that we are going to look at is the genetics. Is it a simple recessive dominant? Is it codominant? What are going to be the impacts of those? Okay, so let's look and let's use the simulator now to investigate and simulate many of these things we've just talked about. All right, so I'm going to click on this link. You can do the same thing if you want and kind of go back and forth, or you can just watch and then play around with a simulator later. Okay, so here we are. Now, just to refamiliarize you with this, let's make it as large as we can. With this simulator. These are the default settings. At a population of 200, we're going to have lots of genetic drift. So if I graph that, I'm going to have lots of random bouncing up and down. And we want to eliminate that because we're only going to look at the effects of natural selection. So I'm going to make this just a really, really big number. So now when we do this, If we have no natural selection, we're going to see the same result simulation after simulation. It's going to have no change at all. So now we can just isolate and just focus on the natural selection. Okay, so let's talk about those three things that we just listed first, right? So first we're going to look at fixation. So let's say we have a big A allele that is best overall. So we'll assign it a relative fitness level of 1. The heterozygote, we're going to look at some lower amount. And then the homozygote will put even lower. So only 80% as much fitness if you're a homozygous recessive, 90% fitness if you're a heterozygote. This is, in effect, a kind of a codominance. We don't have complete dominance here. We'll look at that in a minute. Excuse me. So if I start at 50-50, which is the default setting for the simulation, it's going to take me about, we're looking at 200 generations. Let's just, we'll increase this. Or actually, let's decrease it. Let's do 100. That'll make it just a little easier to determine here. So I have 100 generations. It's going to take a little over 50 generations before I fix that big A allele, which is the most advantageous allele in this population. But if I started at a rate of 80%, right? So 80% of the big A allele, that's where I start. And then there's some change in the environment that suddenly makes something that was in balance, makes it out of balance. There's natural selection. Then we reach fixation a little more quickly. And of course, if I start at 90%, we're going to reach fixation a little bit more quickly, right? So the initial rate of fixation is going to have an impact. So let's go back to 50-50 and we'll reset this. Sorry, I said the initial rate of fixation. What I meant, it was the initial frequency of the advantageous allele. Okay, let's go back to 50-50. The strength of selection is going to play a role also. So let's run it again, just so we have a reminder of what it looks like at this strength of selection. What happens if now the advantage for the big A allele is even stronger? We'll make it quite extreme here. You're only 80% as fit if you're the heterozygote, and you're only 60% as fit if you're the homozygous little A allele. So we reach fixation much quicker. And the stronger this advantage is, the faster we reach fixation. Now, one of the questions in an earlier one, which we didn't explicitly answer, but we talked about the details, let's explicitly answer it here, is can a small fitness advantage have a big shift in phenotype? And the answer is yes, but it greatly increases the amount of time if we only have a small advantage. So if I have a little tiny advantage, let's say we've got 95% is good there and 90% is there. It's going to take, even after 100 generations, we haven't completely reached it, but we still have had a fairly major shift in phenotype, right? We've gone from this nice evenly balanced distribution to, 98.8% of the individuals being homozygous for that dominant allele, and only a small amount of heterozygotes left. Okay, so small fitness differences can make big shifts in phenotypes, but it might take many generations. Whereas if the environment changes to give us a very big advantage, let's make it very extreme, then boom, we reach fixation, a massive change over, this is only like 8 generations. We had this massive change in phenotype. Okay, so let's look at number 3 now, genetics. And some of these genetics can also give us the impacts of overdominance and underdominance. Okay, so let's look first at what would happen with overdominance. So I'm going to clear everything. Let's just set it back to the defaults. Okay, we're going to make our population very large again, so we don't have any confusing influence of genetic drift also acting on this. And let's look at overdominance. So with overdominance, our most fit phenotype is going to be the heterozygote, and our homozygotes are going to see some lesser value. So if both of the homozygotes have the same fitness level, then natural selection is going to keep it right at that 50-50 level. And even if we started at a different frequency, the balance point is 50-50. So if we start at a different frequency, we're going to very quickly reach that balance point, and the natural selection will remain there. And that's why this is called stabilizing selection. No matter where we start out, we're always going to reach that balance point. Okay, so again, this is overdominance. Now, if we change the fitness level of one of the alleles, we're still going to reach a balance point, but we're going to have a different balance point. And no matter where we start here, we end up at that balance point. But the balance point depends on the fitness difference between the two homozygotes. But we're always going to maintain at least some level of diversity in this population. That's overdominance. Now let's look at underdominance. Now in underdominance, it's the opposite. So let's say that both of the heterozygotes have a fitness level of 1, and the heterozygote has a lower fitness level. Let's say 0.8. Now, a lot of people say, oh, we should maintain diversity in that population also, but in reality, we don't. What ends up happening is, depending on which side of this balance point we are, and if the fitness level of the homozygotes is identical, the balance point is going to be right here. Now, because we're starting right on the balance point when we do this, it's going to take quite a while before just a little tiny bit of genetic drift puts it on one side or the other, and then if it does, then natural selection will take over. And so about half the time, and when it occurs is going to vary also because of that impact of genetic drift. About half the time we're going to go up, and about half the time we're going to go down. And if I ran 1,000 simulations of this, I'd get pretty close to 50-50. With small sample size like this, we might be a little bit biased. Okay? So, but eventually, if these populations are competing against one another, we're going to fix one allele or the other. So that is underdominance. And that balance point can vary, right? So if we have a higher level here, so this is still underdominance because the lowest fitness level is in the heterozygote, we're still going to fix one allele, but my balance point has shifted. And so if I get below that balance point, down to 0.2, it's going to fix the little a allele. If I am above that balance point, it's going to fix the big A allele. And you can kind of experimentally test where that balance point is by doing different numbers and seeing which direction it goes. I think the balance point for this one is actually 1/3 or 0.3333. Let's see. Yeah, see, that stays balanced for a long time before it goes, and it's almost always going to go in one direction because I haven't put enough 3s in there. Okay, but if I get above that balance point, I'm going to go the other direction. Okay, so again, that's underdominance. Now, one last thing I want to show you with this simulator, and this is the impact of dominance versus underdominance. And these are all directional selection, so we're just simply going to do directional selection, right? Where the most fit phenotype is 1, but with complete dominance, we're going to have these guys, because it's a complete dominant system, the heterozygotes will have the same phenotype as the homozygous dominant. And so if we get something like this, this is a good simulation for directional selection, but with complete dominance. And so notice, after 100 generations, we haven't fixed the allele. Sorry, this is 200 generations. Let's do 1,000. What happens after 1,000 generations? So this is going to take longer because we're simulating 1,000 generations. So notice the frequency of the big A allele rises very, very rapidly, but then we get closer and closer and closer and never quite reach complete fixation. Okay? Now let's look at the reverse of this. Let's assume that the recessive allele actually has a fitness level of 1, and that if you have the dominant trait, that you have a lower fitness level. Now remember, if it really is dominant, then these heterozygotes are going to show the same fitness level as the homozygous dominant. So we need to put a 0.9 in there. So this becomes a good, and let's just go back to 0.5 as our starting point. This actually becomes a good simulation for a directional selection, but where the recessive allele is the best. And so we're going to try and fix this recessive allele. Now remember, when we were trying to fix the dominant allele under the same conditions, it took over 1,000 generations and we hadn't even fixed the dominant allele. But let's see what happens with recessive allele. After just, this is 1,000, so after about 80 generations, we have completely fixed that recessive allele. So let's put them both back here on the same chart. So there is trying to fix a dominant allele that's beneficial, and this is trying to fix a recessive allele that's beneficial. And notice the difference in the curves. Now, anytime you're trying to fix one allele, fixate it, make it 100% of the population, you can also think of it as removing the other allele. So if it's more helpful for you, you can think of this light purple graph as trying to remove a recessive allele that's detrimental. Right? That's the same thing as fixing a dominant allele that's beneficial. And this is trying to remove a dominant allele that's beneficial. So because of genetics, it takes many thousands of generations to completely remove a recessive allele that is detrimental. But dominant alleles that are detrimental get removed very, very quickly, and we fix the beneficial recessive allele. And so the consequences of this is that natural selection cannot remove recessive alleles very quickly from a population if they are bad. And so remember, in an earlier discussion, we said every individual carries a few recessive alleles that are bad, maybe even fatal. That's because those recessive alleles are not exposed to natural selection very much when they become rare. One last little thing you can think of, here's another way that this helps you to think about this. Let's say you carry a recessive allele that is lethal, but you have the dominant allele that's fine. Is there any fitness cost for you carrying that recessive allele? Now, the answer is to you, no, right? There's no fitness cost directly to you. But remember, fitness depends not only on you, but also on your offspring. How many of your offspring survive? And so if that recessive allele is very, very common in the population, then that can be a high cost because there's a good chance your mate is also a carrier. And then 25% of your offspring will die because they will be homozygous for that recessive allele of which you and your mate are both carriers. But as those individuals die out, that recessive allele will become more and more rare. And so we may reach a point where it's only 1 out of every 10,000 individuals carries a recessive fatal allele. And the chances then of your mate carrying that same recessive fatal allele are very, very low. And so as the recessive allele becomes less and less common in the population, as we move to fixate the dominant allele, which is beneficial, the recessive allele, in essence, can hide behind the dominant allele, and there's very, very little cost of being a carrier for that recessive allele because there's almost, 1 out of 10,000, a very, very low chance that your mate will also be a carrier. All right, I know that's kind of a long explanation, but it's important that we go through some of these details. Hopefully that will help it to stick in your mind and you'll remember it a little bit better.